till now, we saw that bacteria takes a viral DNA copy into the DNA archive called as CRISPR. So, the bacteria scans the entire cell for the viral DNA. When it finds a 100% perfect match, it activates and cuts off the viral DNA, making it useless, protecting the bacterium through the viral attack. What's special about the Cas9 is that it is very precise, almost like a DNA surgeon. What the revolution began when scientists figured out that CRISPR system is programmable.
Decades in CRISPR.
In few decades, CRISPR therapy might cure HIV. You know, that's something very interesting. HIV cures, right? And the other retroviruses, the viruses that hide inside the body, the human DNA-like herpes could be eradicated this way. CRISPR could also defeat one of our worst enemies, cancer. This is a cancer cell. Cancer cell occurs when refuses to die and keep multiplying with concealing themselves from the immune system. That means they hide themselves from the immune system. CRISPR gives us the means to edit the immune cells and make them better cancer hunters. Getting rid of cancer while eventually getting just a complex of just a couple of injections of a few thousands in your own cells, in cells of the embryo, will save you from the cancer. Then we ensure that the cancer is cured and the HIV kind of diseases are also cured with this kind of therapy of CRISPR-Cas9 protein. The first clinical trial of CRISPR concerned treatment of the human patients was approved in early 2016 in the US. Not even a month later, Chinese scientists announced that they would be curing lung cancer patients with this technology. Things are picking up pace very quickly, guys. Next, please. Genetic diseases are very much to be known about for the color blindness, hemophilia, and the Huntington diseases. These diseases occur because there is one cell or one minute change in one minute wrong cell in the DNA archive of the organism. If we cure this one cell, if we correct this unencoded, unappropriately coded one cell with the help of Cas9 protein, we can cure more than 3,000 of the diseases, such as color blindness, muscular dystrophy, etc. So I would like to hand over the mic to Gaurav Ratna sir. He will tell you about the case study when this technology was used in the real human embryo about the Chinese scientists.
So, as you know, what is CRISPR babies and CRISPR-Cas9 protein, what it does. So, we'll talk about the product of it. Product is called designer babies. It's nothing but genetically altered embryos. So, we have the one and only research over here regarding this. It was done by Ha Jiankui, the Chinese scientist, or as he is known as the Mandarin maniac. So, what he did was he wanted to make embryos with the help of CRISPR-Cas9 proteins, which are resistant to HIV, and he did so by genetically altering embryos in embryos. So the objective was to use CRISPR-Cas9 to alter CCR5 gene. Why CCR5 was used? Because CCR5 has in some individuals a natural genetic mutation, which it helps in getting a less susceptibility to HIV. So he used that gene in particular, and then he used embryos. He did the genetic alteration in embryos because at that point somatic cells and germ cells are not divided. So when he did the experiment and was hoping that not only the individual but also his future generations are cured of HIV. So after that, what he did was in November 2018 he announced that the twins were born. The twins, which he could not disclose, of course, and he did so after taking 3 to 4 years of AWOL. And yes, he did not respect the scientific community for doing that. As you can see, there has been such an ethical controversy and such a big dilemma about this. So after that, he did so, and he was also restrained in China for violating ethical problems and understand, right? No, he had zero regard for human life and human genes. So he did so, but he still says he's proud of what he did, and he did for the betterment of humanity. So there's an ongoing debate about this, as you can see. Yeah. Now we'll give my to concluding.
Now, I would like to conclude our PPT on the basis of pros and cons that can be seen in our whole topic. So, what are the cons basically present here? Delivery and efficiency limitations. Can you please read the cons slide? Cons slide. So, the first cons is delivery and efficiency limitations. Successfully delivering into cells is challenging, requiring pressure, concentration, and proper timing. Our next cons is that off-target effects. Off-target effects basically is the unwanted alteration in untargeted genome section, prove significant challenges, difficult to predict and migrate. Another one is that unforeseen sequences. What will be the unforeseen sequences can be present here? Like unexpected changes. CRISPR could lead to unforeseen changes like alteration such as large deletions and duplication in DNA with unpredictable and likely adverse effects. And the last one is the cell types and immune system affected. Basically here, the CCR5 receptors and are not only T cells of HIV lead target, but also on crucial immune cells like macrophages, dendritic cells, eosinophils, microglia cells, cancer cells. Disabling CCR5 has many effects on the immune system, mainly normalizing the function of the human body. And on the other hand, we have pros also. What are the pros? The first process is fast and flexible design. It requires gas release and guide RNA for efficient gene editing. Another one is the multiplex editing. The multiplex editing, because in multiplex editing, we can just simultaneously edit multiple genes at a single time by using different single segregated RNAs that are present in our whole body. And the one of the most important pros of this PPT is that it is one of the most cost effective design because it is simple design reduces cost compared to alternative methods just like Xelevens and talents. Another evidence from the Berlin as a pro we have that while the absence, I would like to submit those pros and cons in the Russian language points. Okay, so evidences from Berlin. While the absence of CCR5 protein is not completely protective, it can lead to significant reduction in HIV infection, which is uncurable. Which can be seen in Timothy Brown, a patient from the Berlin, received transplants from a donor with a virus with a genome of CCR5 delta 32, which is a mutation allowing Brown to stop antiretroviral drugs without recovering from the HIV. So I would like to end up this PPT by just saying over there is a number of practical and ethical changes must be overcome before this potential can be realized at bedside.
Basically, CRISPR-Cas9 technology is a revolutionizing technology to treat many pediatric diseases, and this is going to bring a revolution to the industry.
But still the catch is where does it stop? What are the boundaries? Thank you. Any questions from anyone? Yeah, go to the.
If we unintentionally edit this gene pool of human genome, then what will be the future of the origin of our genome? Will it be zero or will it be converted to some other way?
Unethical is just because it's a human gene, and there are some regulations regarding putting human safety first. There are no such regulations regarding mice or monkeys. So unethical and ethical is just a philosophical dilemma, but it won't affect how we change the genes or how we use the CRISPR-Cas9 protein or the method itself.
Whether it will help or actually affect the evolution of human.
I don't think it will help to use that unethically because you can't review your peers with this. You'll have to do it in secrecy and zero transparency, and because of that, your claims might be disregarded. The efficacy of the procedures might be disregarded, and there is no safety for the human life which can be handled over there.
Excuse me. I have two questions. First question, can you explain CRISPR in simple words? What is CRISPR? and CRISPR technology. Two questions. First is what is CRISPR and explain in simple words because most of students don't know what is CRISPR technology. And next, last question is why CRISPR is so controversial and bad effects.
Thank you. So we will answer this question.
Okay, so as I explained about the CRISPR technology, it uses a protein called as Cas9, which is a molecular scissor, right? So we use in the genetic engineering to cut out like linear ligase is the enzyme which everybody of us know, but Cas9 is a protein I don't think many of us know, right? So it is a molecular scissor basically which can be customizable in our own terms. We can customize this protein, we can put what kind of gene do we really want to cut or we want to mutate, we can put it into the customization slot and then we can customize it. We can change a proteins or maybe a human's or an organism's complete genotype by changing this kind of Cas9 protein. CRISPR is a technology and it is a gene which is basically process name of the process and the gene which is used in this kind of technology. So the next question it was why is it controversial? See, talking about the ethical dilemma, these controversies are only because of the ethical and non-ethical dilemmas. People are happy that there is this technology, but most of the people are arguing about this technology only because this technology has the context of experimenting on humans. People are making lots of fuss about it because this experiment was conducted on a human by Chinese scientists, which was told by them. And only because of this thing that we are experimenting on humans before experimenting on smaller level organized organisms. This is very controversial. Thank you. And then the CRISPR conformance clustered regularly in discrete short palindromic sequences. Thank you.
Any else? Anybody else? Questions? Julie? No? Thank you.
Thank you so much, Gaurav, for that answer. Our next researcher is from Kazakh National Medical University named Dr. Raspendiara, Bonjar Anesthesia and Alexandrovna. Sorry if I mispronounce your names. Their topic are exploring the clinical, genetic and therapeutic dimensions of Hydradrantis suprativa in the arm foot, a comprehensive investigation into a unique dermatological challenge.
Good afternoon, dear professors and students. We're on the third course students from general medicine. Today we're going to talk about the clinical, genetic, and therapeutic dimensions of hydrogenase suppurativa in the armpit. Next, please. Next slide. Next. Next slide. Thank you. So, what is the hydrogenase suppurativa is? It's actually a chronic progressive inflammatory disorder of follicular evolution that presents as painful inflammatory nodules, sinus tracts, and tunnels in appropriate glandular shreds, such as axilla. So this disorder is associated with the low quality of life. So as we can see in a clinical case, here a 45-year-old obese male patient presented with sinus in the axilla. We can see it in image B and C here. So some of his lesions developed in the sinus tract over the time, and he also had a long-standing history of acne lesions on his upper back and the waistline. So the diagnosis of hydrogenase suppurativa was made, and he was actually treated with antibiotics, but it wasn't effective. So unfortunately, he continued to have several cases of hydrogenase.
hidradenitis suppurativa, sometimes called as acne inversa, affect millions of people worldwide. As you know, hidradenitis is a very complex disease that can